So today I'm going to be giving a brief overview of cancer biology. I'm going to begin by discussing what is cancer. So cancer is when there is an abnormal growth of cells due to changes in gene expression, and so these changes can lead to abnormal cell division, proliferation, and cell death. These abnormalities are what causes invasion and metastasis, and there are multiple kinds of cancer that are differentiated by their age of onset, growth rate, cellular differentiation, invasiveness, metastatic potential, response to treatment, and prognosis. So basically, what happens is altered gene expression can be caused by multiple different mechanisms, such as mutation, deletion, loss of heterozygosity, or even abnormal gene transcription or translation. And neoplastic cells that are developing can be developed anywhere within the body, and they can either be benign or malignant. So the first differentiation I'm going to be discussing is how we can identify malignant tumors from benign. And malignant tumors basically can metastasize through lymphatic or blood vessels, and this is what allows them to reach other lymph nodes and tissues in the body, so they don't stay confined to one area. They spread throughout the body and metastasize. Benign tumors grow through expansion and they won't invade surrounding tissues, but they can still become life-threatening by pushing on the normal tissue, and this pushing also pushes on the blood vessels or nerves, and they also have the potential to secrete biologically active substances that can be harmful to the body. So, to further differentiate cancer cells, I'm going to be discussing oncogenes, which are stimulatory, and then I'll later discuss tumor suppressor genes, which are inhibitory. So first I'm going to be discussing oncogenes and how they're relevant to malignancy, their mechanisms of activation, and their biochemical functioning. Proto-oncogenes are genes that can code for proteins, and they help in functioning with the cell cycle, and the name basically indicates the potential to become activated into an oncogene. So originally discovered through studies of retroviral tumorigenesis, the most common mutation by oncogenes is gene amplification. So this results in an overexpression of normal protein products. So I'm going to give you a few examples of gene amplification by oncogenes, and this includes CDK4, which functions in cell cycle regulation, and this can produce sarcomas or familial melanomas. The next gene amplification I'm going to discuss is c-Myc, and this functions as a transcription factor, and when it becomes mutated, it produces lymphomas and sarcomas. And the last one that I'm going to be talking about is ERBB oncogene families, which includes basically HER1 through 4 or HER1 through 4, and these function as growth factor receptors and transcription factors and can lead to multiple kinds of tumors such as gliomas, sarcomas, and osteosarcomas. So while oncogenes can use amplification, they also can lead to excessive amounts of protein production due to translocation, and this happens if there's a highly reactive promoter gene that is placed next to an abnormal gene. So basically the promoter gene just keeps amplifies this abnormal gene and just keeps producing it. Oncogenes can also happen when there's a point mutation, and this is when there's a change in a single base pair, and this mutation could lead to an actually more effective than normal protein, so it can make it resistant to degradation and can produce it in excessive amounts. That can happen when there's a point mutation on the promoter gene, and that's what can produce the excessive amounts of a specific protein. So those are three mechanisms that oncogenes can use to produce cancerous cells. Now we're going to talk about tumor suppressor genes, and tumor suppressor genes, basically when they're normally functioning, they inhibit cell proliferation. So the most common mutation within tumor suppressor genes is point mutation. Tumor suppressor genes can also undergo missense and nonsense mutations. And now I'm going to give you some examples of these tumor suppressor genes and the syndromes that they can cause. So E-cadherin, or CDH1, they regulate cell adhesion, and when they become inhibited, they can cause stomach cancer. P53, which is critical due to its involvement in the cell cycle, they help maintain and control the cell cycle. They are in charge of apoptosis, or can promote apoptosis, and they maintain genetic stability basically. And it's because of this protection from the P53 gene that active oncogenes such as RAS and MYC cannot create tumors by themselves alone. So that's when loss of heterozygosity comes in, and inactivating a second allele at a particular genomic location is what will basically cause the characteristic of tumor suppressor genes to not happen. So basically, P53 is very important, and this single gene is mutated within about half of all cancers. So basically, when the P53 gene isn't working, it is critical to the body, so you'll most likely be diagnosed with cancer, unfortunately. So when a cell is defective in P53, they can escape apoptosis, they can carry out DNA replication of DNA that hasn't been repaired of damage and may have breaks in it, and this allows for amplification of a disrupted genome. Finally, P53, I forgot to mention, when inactivated, can cause sarcomas, breast and brain tumors, and adenocortical tumors. BRCA1 and BRCA2 are responsible for transcription factors and DNA repair, and when they become suppressed, they can cause a multitude of cancers, but mostly they cause breast, ovarian, and pancreatic cancer. So next I'm going to be discussing cancer progression. So in order for cancer cells to invade and metastasize, they must go through a multi-step process. This process is known as invasion metastasis cascade complex. So first, invasion of the basement membrane and cell migration will occur. Then intravasation occurs into the surrounding vasculature or lymphatic system. The third step is being able to survive in circulation and be transported. Then extravasation occurs from vasculature to a secondary tissue. And then lastly, colonization at the secondary tumor site is what eventually creates metastasis, full metastasization. So cancer cells have the ability to recruit blood vessels into the tumor masses, and this is known as angiogenesis. And angiogenesis involves interactions between cancer cells and mesenchymal microenvironments. So some tumors have the ability to produce angiogenic growth factors, for example, VEGFs or vascular endothelial growth factors. They can cause paracrine stimulation of endothelial cells and induce neoangiogenesis and even lymph angiogenesis and contribute to tumor progression. So now that we've discussed all of the different kinds of cancer, sarcomas, sarcomas, I'm gonna explain what each of those means. So sarcomas basically begin in the skin or tissue that line the internal organs, and sarcomas, although they sound similar to sarcomas, they develop in the bone, cartilage, fat, or muscle and connective tissues. So leukemia is a different kind of cancer, and that kind of cancer begins in the blood and bone marrow. Lymphoma starts in the immune system, and central nervous system cancers develop in the brain or spinal cord. So these different cancers, I'm now going to discuss a little bit about how they're treated. So the three main types of treatment are surgery, chemotherapy, and radiation. So surgery involves directly removing the tumor. Chemotherapy involves using chemicals to kill the cancer cells, and radiation therapy involves using x-rays to kill the cancer cells, and oftentimes you'll see that multiple different treatments are used to kind of attack one type of cancer. You'll see that some people want to undergo surgery to take out the bulk of the cancer, and then they may undergo chemotherapy to kind of finish killing off the cancer cells. So although cancer biology is very intricate and definitely will not be able to be summarized in 10 minutes, I hope that this gave you a bit of an overview of what I've learned in this course over these past 8 weeks. And thank you for helping me learn these things. So I appreciate it.